right, so then we move to the next talk by YH Taguchi. Can you share the screen? Okay, great. So we have a talk from Dr. Taguchi. He will be talking about kernel tensor decomposition-based unsupervised feature extraction applied to bioinformatics. Okay, yeah, Dr. Taguchi, please.
Okay, thank you very much for nice introduction, Chairman. And I'll share my slide by chat, so people can now access my slides from chat. I have just sent you a link. And my name is Taguchi, and came from Tokyo, University of Tokyo, Japan. And Chuo University, Department of Physics, Tokyo, Japan. And I would like to thank the organizer for providing me this opportunity to talk about my recent research in this excellent workshop. And the purpose of this talk is just a moment. Yeah. Purpose of this talk is feature selection with large P, small n situations. So in bioinformatics, the number of samples is typically less than the number of features. So here, the number of features is the number of genes, typically it's 10-fold. On the other hand, the number of samples are typically the order of 10 because of the number of human patients. Then why the feature selection with large P, small n difficult is because in the conventional approach, we apply the statistical test to individual feature. Here the two class and attribute p-value to individual feature to reject null hypothesis. Typically, null hypothesis is there no decision between healthy control and patient. This is healthy control, this patients. Then obtained p-value must be corrected because we have too huge number of features, and if the p is huge, the p-value is small as 1 of p can occur accidentally, and we have to correct. Then, sorry. Then features with small p-values, corrected p-values less than some threshold values we selected. So typically this threshold value 0.05. But if number of samples is small, so we can get small enough p-values. On the other hand, so the number of features is large, p-values are heavily corrected, and this means the p-values become larger, and this means less significant. So adjusted p-values are hard to be less than the threshold values. Then one may wonder why we don't use a more advanced approach. Unfortunately, I don't have enough time to discuss about the advanced approach. For example, if we consider Lasso is a famous feature problem method, having a penalty term to reduce the number of features selected. But if we try the Lasso to this kind of problem, we can find a very unstable result. This means every time we apply the Lasso to different samples, we can get a different set of genes. Biologically, it is very difficult because we need some specific set of genes to some specific targets. So if every time we apply the Lasso to different samples, we get different set of genes, it is useless. Then our approach is very different. First, we try to find some sample vectors which is consistent with the desired property. For example, this is distinction between healthy controlled patients. And then selected features whose contribution is large to these sample vectors. And this contribution is projection is assumed to be of a Gaussian, the p-values are attributed, and the features having larger contribution to sample vectors selected. So this can be as follows. Suppose we have n samples, so n samples composed of the healthy controlled patient. In this space, each individual feature, the typical genes, having n gene expression of n samples, then it can be regarded n dimensional vectors and these points in n dimensional space. If I have p features, then it composed with cloud, and if we project the gene expression of the individual feature to each samples, we can have this kind of distribution here. This is the samples distribution for p genes. Then if we occasionally have some sample vector which distribution distinct between healthy control and the patients, then we can project each individual vector's future into this projection, and this projection has some distributions, just like so Gaussian, and we assume that this prediction assume Gaussian distribution, we can add p-values. Then some outstanding features having larger contribution to this prediction selected. So the problem is how can we derive these sample vectors? We employ tensor decomposition. The reason why we employ tensor decomposition because the measurements often associated with multiple characters. For example, the measurement has been done healthy controlled patient and for tissues. Then ICE genes having two suffix G and K, and it is represented as tensors like this, the genes and some person tissues. Then we apply a tensor decomposition. So I don't have enough time to explain what a tensor decomposition is, but we can get sample vectors attributed to patients, sample vectors attributed to tissues, and projection to these sample vectors. Then mathematically, this is written like this. This is a unit vector, this is weight, and summing up to come back to the original tensor decomposition, tensor which represents the expression. Then these two sample vectors which you would like to have, and this is projection to these sample vectors. So it is automatically you get combinations sample vector and the projection vector. Then now I would like to apply this method to the drug differentiation in COVID-19, the Major just explained to us. So this has been published in last September. And since then, this paper accessed 5,000 times. And if you are interested in details with this part, you can read this paper later. Then the gene expression profile I studied in this study, the number of genes is this 20,000. We have 5 cellular lines. This is infected and non-infected. And for these combinations, we have 3 values to replicate. Then the gene expression of I express the tensor like so this is the number of genes, this is the number of cell lines, this is the condition if infected or non-infected, this is biological replicate. Then we apply tensor decomposition to tensor decomposition, and we can get this, and this is a sample vector for cell line, the sample vector for distinction between infection and non-infection. This is the biological replicate sample vectors, and this is projection vector of gene to these sample vectors. So then this is a heavily large P small problem because the number of features is tenfold, while the number of samples are small is only 30. So this is the ratio between the number of features to sample is 1,000, and this is a very difficult problem. Then the sample vector desired should be independent of cellularly and virus replicate, but this should be distinct between the infection and non-infection. So this is the result of tensor decompositions. The first sample vector to be the cell lines has almost constant value of cell lines, and the second sample vector to be to the infection and non-infection has distinction between infection and non-infection. And the first vector to be to the virus replicate has constant values. We decided to employ these sample vectors. And persistent sample vector can be found by which G has largest weight. Then we have found that these sample vectors has largest contribution to fifth projection vector, and we have employed gene vector which predicted these 3 sample vectors, and we assume the p-values. We assuming U5Y obeys Gaussian distribution, and p-values are attributed to this chi-scale distribution, and p-values collected by Benjamin Horville method, and finally you have gotten 166 genes with outstanding very small p-values even after we collected this and among this large number of genes. Unfortunately, I don't have enough time to explain the full outcome, but I would like to introduce one outcome. So we have checked if the selected 106 genes are overlapping with the human genes which are known to interact with SARS-CoV-2 protein. This is the SARS-CoV-2 proteins, and this is p-values by computed by Fisher's exact test. As you can see, almost all SARS-CoV-2 proteins are 163 genes are interacting, overlapping with the human genes which are known to these SARS-CoV protein. So this means our strategy successfully identifies limited number of 163 genes which are very critical to SARS-CoV-2 infections. If we apply the conventional approach, it's terrible. For example, we apply the t-test and some we cannot have no genes and on the other hand we apply RIMA, almost all genes are distinct. Then there's no way to select the limited number of genes which is used for drug discovery. Unfortunately, I don't have time to explain the drug discovery part, but the people can read this paper later. Now we would like to this strategy extend to kernels. So the advantage kernel is because we can read the size. So originally we have to have this size of tensor the p times n1, n2, n1, n2 is the number of samples. Now using a tensor kernelized, we can have only this size of tensor. If n1 and n2 sample is smaller than number of features, it is great reduction of the space. This part has been published this May, this journal. So if you are interested, you can read it later. And previously, we have directly applied the tensor decomposition of this tensor. This is the number of genes. This is 2 samples. So this is in the SARS-CoV-2 cases, this is the number of cell lane and biological replicate and so on. Now we can apply the production is the tensor summing up I, and we can do this computation. That this resulting tensor is very small because this tensor has size of this. So then one N2 is the order of 10, and this is very small. And we apply the tensor decomposition to this small tensor. We can have sample vectors. Then this is mathematically can be written like this. And we also replace this part with any kernels because this has the form of linear kernels. So for example, we can replace this linear kernel with larger base function kernel, polynomial kernels. So we can kernelize this strategy. Then for future section for linear kernel, we can easily compute a projection vector for this sample vector. So we can do p-values and we can select limited number of genes similarly. But for kernel cases, we cannot take prediction. Instead of that, we can remove some specific I and compute sample vector repeatedly. And if which future has drastically reduced the coincidence between the sample vectors and the distinction between the infection and non-infection. And we have top genes who affect who destroy the coincidence between the sample vectors and the infection and non-infection. So I don't have enough time again to explain this, but this is the result of tensor equation just I have introduced. And the kernelized one has more advanced result because the p-values which is computed by the Fisher test, which estimate the overlap between the selected 160 genes and the human genes which not to be interact with SARS-CoV-2. It is clearly improved. So kernelized can improve the selection of human genes which is more coincident with the human genes, which is known to be interact with the SARS-CoV-2 protein when infection takes place. Finally, I would like to apply this strategy, the integration to the multi-omics dataset. So this is published in this preprint. If you are interested in, you can read it later because I have uploaded this slide through the chat. I have just sent you the link of this slide. I have integrated in this paper so 4 distinct multi-omics dataset, methylation, the gene expression, the 2 proteome. And as you can see, the number of features very different. So for methylation, this is the microarray dataset. The number of probes this number, and for the proteome, we have very small number of features and the number of genes in between. It is not easy to integrate genes for omics database, but if we apply the kernelized method, it's very easy because you can start from this tensor. PK is very different from these for omics dataset, but we summing up PK, the resulting kernel has same forms, N1 times N2, then N2, and we can bundle this for tensor into this file. Now the K is the number of omics here, the 4. And we have N1, N2, N1, N2K tensor, and this is very small. We can apply tensor decomposition. We can have sample vector, and the prediction can be computed this, and we can again attribute p-values to these features, and we can select. Again, sometimes I don't have no time. to explain the fully the result of this result. But for example, for methylation case, although the number of probes, this number, our method can correctly select the limited number of methylation probes, and this genes are associated with these methylation probes. And I have found this limited number of tensor transcription factor targeted genes, and this transcription factor has useful biological meaning. So our strategy can be successfully applied the integration to multi-omics data set. So conclusions. So we have invented the kernel tensor decomposition based up feature extraction, and it can reduce the required data size from P to N. The P is a huge number, but we have now only small size tensor whose number is 10 times 10, something like that. And it can also integrate the multiple data set. Individual omics data sets has a very variety of the number of features, and it is very useful method. Thank you very much for your interest.
Okay, thank you very much for the presentation. So any questions from the audience? We have a few minutes.
Can I ask a quick question?
Yes, thank you.
Very nice talk. So you mentioned that if we use, let's say, a t-test to identify features in cells that are infected versus non-infected, and try to correct for p-value, we are getting very few genes or no genes. And then that's correct, and I understand that. However, generally, that's not the practice that is being used for identifying differentially expressed genes, right? So we generally take a negative binomial distribution of the reads that are different between two conditions. And that's, so have you compared it with the more kind of common things that is being used, i.e. the tools that are recently being used for identifying differentially expressed genes?
Very nice question. I think, for example, you mean using D6 HR.
That's correct.
Yeah. But firstly, if we would like to use this kind of method, we have to have a sequencing data set. But my method is not restricted to the sequencing data set, because the last example, I have integrated the sequencing data set and micro data set, because this is multi-omics data set. So in this sense, for example, in this last case, we cannot use HR D6 because methylation data set is not a sequencing data set. So we cannot use this kind of strategy. And also, I have checked sometimes HR D6, but in some paper I have compared the performance of method and HR and D6. It cannot compete with ours because it is heavily, in some sense, large piece problems. So it is very bad. So unfortunately, I have no example here. But even if we apply the HR and D6, it cannot get good enough results that I can have with my method. Maybe you can trust me.
Thank you.
Okay, we can take one more question. Okay, so I have a very naive question. So from you started from the tensor decomposition and then did the kernel, and then you can get a more significant the p-values.
Yes.
I just naively wonder, but you started from the same data.
Yes.
Right. So and then this kernel considers it's a well, data twice. You multiply it.
Yeah.
Right. So if you multiply, okay, I have a very naive question. So if you multiply it 3 times, do you get more? If you use a kernel which multiplies the data 3 times, not
Mhm.
just 2 times, then
Mhm.
does p-value get more significant? Or
Yeah, okay, I understand what you say, but I'm sure in that case is perhaps yeah, it is. We should check it, but I cannot check it. But me, I don't know how to apply the 3 times, but maybe I will try. So if what you are saying is correct, so I should check it. Sorry, I cannot answer this your question now, but very nice question. So I will try.
okay, okay. Thank you very much. Let's thank speaker again. Thank you very much, Dr. Taguchi.